Okay. All right, so just for the sake of the recording, I'm going to be drawing on this board, so I'm sorry for those of you who have your backs to me, sorry. But I'll be drawing up there. Okay, all right, so we're gonna go ahead and get started. I will unmute this. All right. All right, so we have our types of evidence. This goes back to the very first lecture. We're talking about the scientific method. All right, so can anyone tell me what anecdotal evidence is, or an example of anecdotal evidence? What's an example of anecdotal evidence? Okay, let's say I ask Camille, say, Camille, what, your hair's pretty, what kind of shampoo do you like? And she says, oh, well, I like Dove. I like Dove shampoo because it makes my hair really soft. Okay, that's anecdotal evidence, right? So that's personal, as someone's personal testimony. Okay, so when you think anecdotal evidence, think personal testimony. So do you guys think that this type of evidence would be reliable?
Maybe depending on who's saying it.
Okay, depending on who's saying it?
It's not because it's simple study.
Okay, right, so yeah, if you didn't ask enough people. Awesome, anybody else? Oh, I'm sorry. Oh, okay. Yeah, so you're right, this isn't very accurate because you're asking one person. Also, it's all based on personal experience, right? So just because Camille liked that shampoo doesn't mean that, you know, someone else might tell me that they didn't like the shampoo, okay? So it's all based on someone's personal experiences, so it's not necessarily the most accurate or the most reliable source of information. Okay, so we have our anecdotal evidence. Then we also have our experimental evidence. Can anyone give me an example of experimental evidence? Or how would you get experimental evidence? And this is not a trick question, I promise. Exactly, right? We're running an experiment, right? So experimental evidence is saying, okay, I am testing whether students who study longer get better grades. And I run an experiment and I say, okay, I'm gonna have this group of students study for 2 hours, this group of students study for 10 hours. And the group of students who study for 10 hours have higher grades. I could say, okay, I think that students who study more will have higher grades. But because I ran an experiment and now I have some data to back up my theory, this is now experimental evidence and it's now reliable information. Okay, so pretty much the takeaway is that the experimental evidence, you're running an experiment. Okay, so running an experiment, and it's the most reliable source of data you can have. So that's why in psychology and biology, chemistry, we talk about running experiments, okay? Getting actual data to see if we can support or refute our hypothesis, okay? So in experimental evidence, we're gonna run an experiment. The important thing is that it's the most accurate or most reliable type of data. Okay? All right, so then what's my third type of evidence? Yeah, epidemiological. Let's see if I can. This one's a little bit more tricky, but pretty much you're synthesizing data or information from a large amount of data that's been collected over a long period of time. So let's say it's experiments that happened, not an experiment that you ran during like the initial time, but you're looking at a large amount of data from a past experiment, okay, or from past people. So With epidemiological, the difference between experimental and epidemiological is you're looking at data that's been from the past, but it's over a large amount of people, okay, or a large amount of subjects. Okay. Okay. So kind of epidemiological research, this is more common in psychology, where you're kind of surveying or looking at experiences from a large amount of people over a large amount of time. You're not necessarily running an experiment, right, having controlled variables, independent variables, but you're getting data from a large amount of subjects, and you're able to glean information from that. So epidemiological, that's also very reliable. However, you need a lot of time and subjects to get that. So that's a little bit harder to get than experimental or anecdotal evidence. All right. Are we good with that? Okay. All right, so we'll move on to the next question. All right, fantastic. You all remember these. So this, I would definitely know this for the exam, and you all do, so that's great. In class, you mentioned that that's definitely probably going to be on there. So what does it mean to say that a hypothesis is repeatable?
We can do it.
Right, awesome. In scientific papers, biology, chemistry, one of the big criteria is that other scientists can repeat your experiment and get the same results. Okay, so it needs to be something that if you gave me the formula to make something, like if you make a cake, you should be able to give me the instructions so that I can replicate that cake. Okay, same kind. What about testable? What does that mean?
It can be tested again after the first time.
Yeah, it can be tested again. Also that it could be tested, period, right? What if I said, Bailey, right? Yes, I'm so excited. Bailey, I want you to run an experiment on whether kittens make people happy. What do you think could be a possible problem with that experimental question? Anybody can answer.
It's hard to measure.
Right. What's hard to measure?
Like, people's happiness, it would be really dependent on the person. How would you even know?
Right.
Like, how to...
Perfect. Right. Exactly. We need to make sure we're asking the right questions, right? We need something that we can easily test, right? Something that's not relative. So exactly. What about falsifiable? What does that mean? Right, exactly. Yeah, so we need to be able to have a question that we can prove right, but also that we can prove wrong. Awesome. Yep, so those are the three characteristics of a good scientific hypothesis. So those are the two questions that I picked out for lecture 1. Does anyone else have any other questions about that information? In question 1 about the scientific method? No? Okay. So I am looking at, I've uploaded the outline review topics. So maybe while we're going through, it might help if you see something on the outline that you don't quite get or you want to go over. Feel free to stop me and we'll look at that. All right? Okay, so now we're going to move on to lecture 2, which is about statistics. All right. So what am I asking when I'm saying does correlation equal causation? What am I even talking about?
You know, like things having patterns, like there's like two things that go in the same way, but it didn't cause it, it just kind of happened like that.
Awesome. Anyone else have anything to add to that?
Like correlation would be, do two things have a relationship, but even if they do have a relationship, you can't prove that one thing causes the other.
Exactly, right? Yeah. It's kind of an issue that happens when we do experimentation, right? Just because you see that there might be a correlation between amount of ice cream someone eats and weight gain, that doesn't necessarily mean you can say that eating ice cream causes weight gain, right? So even though a couple of variables may be related, that does not mean that one variable causes another thing to happen, okay? Yeah. So correlation does not equal causation. All right, any other questions about that before we move to the next question in this section? All right. All right, awesome. So I put this question in because for this chapter of the book, the key point was talking about the media and how scientific data can be misinterpreted by the media and also by the public. So I could see maybe that as being a question. But awesome. So media leaving out key details of the experiment. Data perceived as isolated when really it's more complex. There's more going on than just saying that people who, I just said this, but people who eat ice cream gain weight. They're leaving out details, okay? Media headlines are sensationalized, yeah. So these are all ways that misinterpretation of data can happen in the public and also what's problematic. Let me see some other. So also in this section was the different graphs, right? So the linear graph, sinusoidal, logarithmic, exponential. Get them all? Let's see, I think we may have said them all. We said, yeah, but the graphs, right? So if you can at least recognize them, if you saw them, then I would say that's good too. What's the difference between the alternative and the null hypothesis? All right, anyone else have anything to add to that? Yeah, perfect. So I'll write that out. So the null hypothesis is saying that there is no relationship between the two variables, right? So someone throw out an example of an experiment. I think I'll do the grades one. So let's say that my hypothesis is that more study hours, and I'm just going to shorthand this, equals higher grades. Okay? So what do you think the null hypothesis would say? That was my statement. What would the null hypothesis be? I'm sorry?
That it doesn't equal higher grades?
Yeah, so I would say that there's no correlation between study hours and grades. So one won't, they don't have an effect on each other. So number of study hours does not affect grades. Okay? And what about the alternative? What would the alternative hypothesis say?
The more you study, the higher your grades.
All right, any other ideas too? Yeah, gosh. So the alternative hypothesis would say that there is a correlation. And so then that's when you would be running the experiment to see, well, would more study hours increase grades or decrease grades? So null is saying that there's no correlation. Alternative is saying that there is a correlation, but we're not sure which one. So it would be like number of study hours does affect grades. All right. Awesome job, you guys. All right, so is there any questions about the null or gosh, words, null or alternative hypothesis? No? All right. Okay. All right, so that is all the questions I have for lecture 2. Any other questions about the information from that section? All right, let's move on. Okay, great. So this, we're talking about the characteristics of life. 3 of them are there. So what is sense and respond to stimuli? What am I talking about there? What am I talking about when I say sense and respond to stimuli? Like what would be an example to sensing and responding to stimuli?
If you touch the stove and then like put your hand away, so it's hot.
Exactly, right? Awesome. So interacting with your environment, right? Growth, that's probably pretty obvious, right? But how is growth different for multicellular and unicellular organisms? What's the difference there?
In unicellular, they just get bigger. So it's like, but then in multicellular, they cells.
Yeah, awesome, exactly. How did you know that? Okay, yeah, awesome. And another way that you can get there is that for unicellular, right, they're only one cell, so they can't increase their amount of cells, right, unless they're reproducing, right? But to just grow, that means that their one cell would just get bigger. So exactly. But for eukaryotic, they would increase the number of cells, right? Like all of us, we started as one fertilized cell, and then we became what we are now, which is lots and lots of cells. So awesome. Then we have reproduction, right? But there is one that I missed. What is one that I missed?
Taking using energy.
Okay.
Maintain homeostasis.
Maintain homeostasis. Awesome. What is homeostasis?
Just like keeping your body regular, normal, just like keeping like.
Exactly, right? Awesome, thank you. So homeostasis is maintaining a constant environment, right? We talked about, can't remember what system we were looking at. I think it was the nervous system. We were talking about thermoregulation, right? What happens whenever you're cold, what happens when you're hot, what your body does to maintain a constant temperature. So we've learned, right, looking at the body systems, how homeostasis comes into play, how sense and respond to stimuli comes into play, right? Awesome job. So you kind of probably see now, like with reviewing, that with the information you've gotten now, it's really built already on what you knew. So it's not going to be hard to, see this stuff. So great job, you guys. All right, we have one more from this section. Awesome. Okay, why did you guys say water? Why didn't you say soil?
That's all living things in water.
All right. Anybody else? Okay. Awesome. Great job. So water, as you learned, is a very special molecule or compound of the same. Remember we talked about there are certain properties of water that make it special. Do you guys remember any of those properties of water that we talked about in class?
Adhesion and cohesion.
Okay. All right. Anyone else? I'll tell you one. There's polarity, right, in the molecule. Which state of water is more dense, solid or liquid? Which one is more dense, solid or liquid? Liquid, right? That's why ice can float, because liquid is more dense. Let's see. Liquid state of water is more dense. Also, water has a high heat of vaporization. All right. Okay. I think those are all that we talked about in class. Can you guys think of any other ones? Okay. So all of these are connected. All of these are connecting back to the structure of water. Can anyone tell me what the structure of water is? Or what three molecules are involved in the structure of water? Yeah, H2O, right? Hydrogen, what? Oxygen. Sorry. And they're all bonded, so these are bonds. What does it mean to say that a bond is polar?
That it has a partial charge.
Yeah, awesome. So which has a partial positive charge? Okay. Is anyone else have an idea? All right. Yeah. What did you guess?
I just
Yeah, you
more
knew.
than the oxygen.
Mhm. Oh, why did you know it wasn't the oxygen?
I don't know.
Okay. No, you're good. You're good. You know this. So oxygen, this goes back to a property called electronegativity. Has anyone had chemistry yet? Okay. Well, if you do, this is kind of a precursor. This will be one of the first things you talk about. So when you have an element that is electronegative, that means it has a strong affinity for electrons, right? Electrons are negatively charged particles. So if an element has a strong pull on electrons, which are negatively charged, what do you think the charge on that element will be? If the element has a stronger pull on negative particles, pulling negative particles closer to itself, you think it'll be more negative or more positive? Anyone else? Anyone else have an idea? That's okay. This is kind of a so elements are bonded because they're sharing electrons. And so if oxygen is pulling more of the negatively charged particles towards itself, then that would mean that the oxygen would have more of a partial negative charge because it's pulling the electrons, which are negatively charged, closer to itself. So since you're pulling the negative charge away from hydrogen, that means hydrogen would be more positively charged, but in comparison, because the electrons are closer to the oxygen molecule. Okay? This is why you have your partial positive charge on your hydrogen and your partial negative charge on your oxygen. And this property is what causes all of this stuff to happen. So this that we just explained is polarity. Okay? Because of this negative charge, and when you were a kid, you probably played with magnets, right? You learned that negative charges are attracted to each other. Bless you. So imagine we have a cup of water, right? There are a lot of molecules in there, right? So you have a lot of H2O molecules in there. Okay? And so as you can see, when this happens, you have oppositely charged species close to each other. Well, this one again as well. You have oppositely charged molecules close to each other. Okay? And so you can probably see that opposite charges can begin to attract to each other. So this positively charged hydrogen will be attracted to the negatively charged oxygen. This positively charged hydrogen will be attracted to the oxygen. This is what allows molecules to stick together, and that's called cohesion. It's also what allows molecules to stick to other surfaces, that's adhesion. Okay? This also allows liquid water to be less dense as a solid, because whenever you freeze water, what do you know about ice? It's kind of a crystal structure, and that's because the molecules are separating out as far as they can from each other. And so that's why ice is less dense than water. These bonds are very weak by themselves, but you have billions of molecules of water, right, in a sitting. And so that's why there are a lot of hydrogen bonds, which means that the molecules stick together really tightly. And you probably notice whenever you're making like ramen noodles or soup or something, it takes a long time to heat up that water, right? That's because you're having to break all of these bonds, okay? So sorry, that was kind of a long-winded explanation, but if you can remember that a water molecule is polar, and you can remember roughly what the structure is, then you can figure out all of these things. They're all connected to the structure of water, okay? So you'll be able to slay any chemistry question she throws at you if she does. All right, so those were the two questions over the Life on Mars Lecture 3. That's just the basic principles of life. Do you guys have any other questions over that material? Okay. All right, we'll move on to the next question. Okay, awesome. So antibiotics are medications that stop the growth of bacteria. An antiviral medication would be that would attack a virus, okay? So an antibiotic would not be able to stop a virus, vice versa, an antiviral medication wouldn't be able to stop bacteria, all right? I asked this question because this specific lecture was over penicillin. This is lecture 4. So she may ask you a question about what penicillin is, what does it fight, why was it made, how does it kill bacteria, right? Does anyone remember how penicillin kills bacteria?
Something about the ribosomes.
Yeah, so it's disrupting the cellular structure, right, and attacking its cell wall or its, I don't know if you talked about peptidoglycan, but you're right. It has to do with attacking the ribosomes, and so that you're disrupting the machinery of the bacterial cell. All right, any questions about this? All right, we'll move on to the next question. All right, so let's talk about this. So this is talking about the difference between prokaryotic and eukaryotic cells. Can anyone tell me just a general definition, what's the difference between those two types of cells? Is which one? Okay, anyone else have any other, or do you have any other ideas?
Wait, no.
Oh.
Is it prokaryote?
Right. No, you didn't say nucleoprotein. Yeah, exactly, right? So prokaryotic cells, they don't have a membrane-bound nucleus, right? So eukaryotic clearly means new karyote or new nucleus, right? So they are, they have membrane-bound nucleus. Does anyone want to get up and write down some aspects of prokaryotic and eukaryotic cells? Or just one? You could have two students, one do prokaryotic and one do eukaryotic. No? You just want to sit here and, okay. As long as I'm not boring you. I don't want this to be a boring review session. But yeah, so prokaryotic, right? We have not a membrane-bound nucleus. Would you find chromosomes in prokaryotic cell?
Oh, well.
Would you find chromosomes in a bacterial cell? So you would have, I don't know if you went over this in class, you would have circular ones, but. So the big thing to know is that with prokaryotic, you don't have a membrane-bound nucleus. With eukaryotic, you do. Which one has organelles? Eukaryotic, right? Eukaryotic has organelles. Which one, so we said plant cells are eukaryotic. Which ones are animal cells? Eukaryotic. Which one is like bacteria? Prokaryotic. Awesome. Do you guys remember any of the organelles? So we said the nucleus. What's the point of the nucleus?
It has instructions for.
Yeah, exactly right. That's where all of our DNA is held. That's where we transcribe DNA. Awesome. Any other organelles you guys remember?
Mitochondria.
Oh yeah, what does the mitochondria do?
Like the powerhouse.
Yeah. Right? That's where our cellular respiration happens. Right? We're talking about with plants. Well, not plants and animals do it, but we're talking about plants. Yep. Mitochondria, nucleus. Anything else?
Lysosome.
Lysosomes. Awesome. Peroxisomes. Golgi apparatus, right? The packaging center. Endoplasmic reticulum. Yeah. So I don't think you need to know all of those, but just recognizing kind of the bigger ones like the mitochondria, the nucleus, and that those are found in eukaryotes, membrane-bound organelles. All right. So I think that's my last question on the penicillin, the wonder drug section. Does anyone have any other questions about lecture 4? No? All right. All right, fantastic. So you guys all remember that photosynthesis is a process of turning energy, sunlight, right? Well, turning sunlight into sugars, right? Or foods that the plants can use. What about cellular respiration? What's that one? I don't remember that.
Is it like the opposite of photosynthesis?
How so?
Like I know the equation is flipped.
Mm-hmm.
I can't remember what it is, honestly.
Yeah, no, you're fine. You're absolutely right. So cellular respiration is the opposite of photosynthesis. So with photosynthesis, we're using sunlight to produce sugars. But in cellular respiration, we're taking those sugars and breaking them down for energy that the cell can use. So if you think about it, it's like you bake bread and you have bread, but it's not helping you. You have to eat that bread, right, and metabolize it. So that's kind of like cellular respiration. With photosynthesis, we're making the molecule. Cellular respiration, we're breaking it down. What about endosymbiosis? Anybody remember what endosymbiosis is? What that even pertains to? So chloroplasts and mitochondria are special organelles. They're called plastids. They're special because they have their own DNA within the eukaryotic cell, okay? And they're able to carry out their own activities. And we're wondering, how did that happen? You know, that they have their own DNA, they're their own independent entities within the cell. So they help each other, they help the cell, but the cell helps the chloroplast, mitochondria as well. Endosymbiosis is the theory that mitochondria and chloroplasts started out as prokaryotic cells, and they were engulfed by another cell, which used them, and they became kind of formed a relationship together. So this is just a theory for saying how did chloroplasts and mitochondria get into a eukaryotic cell. This is kind of the theory that drives the production of eukaryotic cells. Okay, that was a very minor detail, but if it comes up, you'll know it. All right, so next question. All right, awesome. So, yep. So we know that plant cells, they contain a cell wall, right, outside their membrane bound of their plasma membrane. They are photosynthetic. Awesome. Autotrophic. What does autotrophic mean? Exactly, right? Autotrophic means that plants produce their own food. So the way that plants carry this out is through photosynthesis. Okay, so you can think about those two kind of being related to each other. And we know, right, that plant cells are characterized as eukaryotic, which means that they do have a membrane-bound nucleus. All right, are there any other characteristics of plants you guys can remember? Oh, that's all right. These are the big ones. So these are ones that I would remember for sure. Cell wall, autotrophic, photosynthetic. Those are the big details. All right, so that is all from lecture 5 on plants. Any questions about that before we move on to lecture 6, DNA? No. Okay, well, go ahead and move on. Okay, great. So what is DNA, right? What do we remember those 4 macromolecules that we talked about in the beginning of the lecture? Well, in the beginning of the semester, I guess. Mm-hmm. Okay, so I heard proteins, lipids. What were the other ones?
Carbs.
Carbohydrates.
Monosaccharide.
So monosaccharides would be carb. But this one is actually the class that DNA is in.
Fats.
So fats are lipids. What does DNA stand for? You remember that really long name?
Nucleic acids.
Nucleic acids. Awesome. So these are your genetic material. So nucleic acids. All right. Awesome, you guys. What are some characteristics of DNA? What does it look like? What's the shape of DNA? A double helix, right? And what are the building blocks? So what's the building blocks of nucleic acids? What are the monomers of nucleic acids? Nucleotides, right? So DNA is made up of nucleotides. You might remember what those nucleotides are. So I'll start you off. Adenine, guanine, thymine, one more, cytosine. Awesome. And there's one more. This one is found only in RNA. Starts with a U. Uracil. Awesome, you guys. Great. So these are our nucleic acids. And as you know, right, we have a specific pairing pattern, right, for our DNA and RNA. Do any of you remember what the pairing pattern is? What's paired with what? A and T, C and G. And that's for DNA. What about if I have RNA? A and U. Awesome. Man, I can tell you guys have been studying. Awesome. All right. Any questions about this? All right. Yep, so DNA is the genetic material, double helix made of nucleotides. All right. Awesome. All right, so we'll move on to the next question. Awesome. So what am I talking about when I'm saying that it replicates through base pairing? What does that mean? Well, what roughly happens when DNA replicates?
Transcription and translation.
Okay, so that's for protein synthesis, but you're very close. What has to happen whenever you're transcribing DNA? What do you have to do first?
Doesn't it make like another copy?
Exactly.
I don't know exactly.
That's okay. That's awesome. So we're making a copy. And so in order to make a copy, we need a template, right? So what happens is we have our DNA strand. This is like our parental DNA strand that act like I untwisted it. What happens is we're going to take one of these strands of DNA, let's say this is the parental DNA, and we're going to use that, we're going to use that as a template to make another strand of complementary DNA, and that's through base pairing. So we're going to take the base, or we're going to add complementary bases to the parent strand. So when I say replicate through base pairing, that's what I mean. Did that clear things up a little bit? You guys knew that DNA did that, so that's awesome. But I just wanted to clarify just in case. So those are my two questions for the lecture on DNA. Any questions on the DNA lecture outside of this? All right. We'll move on. Okay, fantastic. So now we've moved on to the genetic engineering and proteins lecture 7. So we're talking about GMOs, right? Genetically modified organisms. What's transgenic? Or transgenic of that? What's kind of the word you hear in there? Gene, right? And trans, or to move, right? Transition. So we're taking the genes from one organism and we're moving it to another. So it's like, say, I think this was in your book, there's an example of a goat whose milk can be used to spin. The milk has the capacity to make webs. So what they've done is they've taken the genes from a spider and they've taken them and added it to a goat so that their milk can produce webbing. Okay, so that's like taking genes from one organism and transplanting them into another. That's a transgenic organism. But they would also technically be characterized as a GMO because you're altering their genetic makeup. Okay? Any questions on that before we move on to the next question? Okay, so we'll go over this. Yeah, so this was one of the driving forces for the, this is what your book was talking about in that chapter, was GMOs and making new organisms. So a virus, right, what is a virus? We talked about that, I think, in just the recent lecture, or the one before that, but what's a virus made of? It's made of a protein coat, right? And what's in that protein coat? A virus equals a protein coat plus RNA, right? And what is the RNA? in here, it's that genetic material. So whenever a virus infects an organism, what happens is the virus will, let's say this still squares a virus because I'm terrible at drawing. And the virus will perch on that cell and will inject its DNA into the cell, right? Kind of like, you know, putting stuff in there, okay? It's putting its DNA into the cell so that the cell, it hijacks the cell machinery and uses the cell to make more of itself, okay? So as you can imagine, if we took a virus and put new DNA into the virus, that virus would then put the new DNA into the cell, okay? So essentially, with using a virus as a vector, you're putting your desired DNA into the virus, then letting the virus infect the cell or put the new DNA into the cell. Does that make sense? Okay. And then with creating a recombinant gene, that's you're taking the regulatory sequence from one organism and adding it to the coding sequence of another organism, okay? I don't really see you needing to know the nitty-gritty details of both, but just recognizing that both of those are ways to make a GMO, I think would be helpful. Okay. Do you guys have any questions about that? Okay. Do you have any questions about this lecture? Bless you. Okay. All right, then we'll move on to lecture 8, stem cells and cell division. All right, awesome. So what is the difference between, well, what are we making in mitosis? Right, so in mitosis, let's go ahead and make another chart. Does anyone want to come up and write what happens in meiosis or mitosis? I want to probably give that opportunity if you want to, but if you don't want to, it's fine. So yeah, mitosis, we're making somatic cells. What do we make in meiosis?
Gametes.
What are gametes?
Sex.
Perfect. So we're making sex cells. All right. In which process are we making cells that are diploid?
Mitosis.
Mitosis. What does diploid mean?
Has 2 sets of
Exactly, right? 2 sets of DNA, right? And so that means meiosis makes haploid cells. That means they have half the amount of DNA, which makes sense, right? Because whenever your sperm and egg unite, they need to be able to make a diploid cell. So they must each have half the amount of information. So they're haploid. Okay? That's kind of the defining characteristics of both. What's a stem cell?
a cell that hasn't been specialized into anything can become anything.
Right. And what's that process of becoming specialized called? Exactly. Yeah, those are both mentioned in this lecture. Anybody have questions about those things? Or meiosis and mitosis? Okay. On your exam that you had over this material, did she have you draw out or recognize certain stages of the cell cycle? She did? Okay. Because I didn't want to take your time on it if she didn't do that. Okay. So here I'm asking you about the cell's life cycle. So mitosis is the replication of somatic cells, right? Do you guys remember seeing a diagram that was circular? And you have mitosis, you have G1, you have S, you have G2, okay? This is your cell's life cycle. Can anyone tell me what the M means that I put here? Mitosis, yeah. So this M means mitosis. And what overarching stage encompasses G1, S, and G2? Interphase. So the answer to this is interphase because you're in the interphase phase of life before you get into mitosis. So interphase is all the time before you actually divide, okay? Can you want to tell me what the G1 section is? Growth, right? And sometimes you'll see some books will have G0, and that just is for cells that never divide. But G1 is growth. That's like the cell is getting ready to, getting ready to start the process of dividing, right? So it's growing, it's making sure it's in a stable place to be able to divide. The S phase is synthesis, and that's where the replication of DNA occurs. So if you get a question that says you replicate DNA during prophase, it's false because you replicate before you get to mitosis. Okay? G2 is growth 2. Okay, the cell is again growing and making sure that it has the right amount of DNA before it divides. Okay, so for this answer, right, interphase is the section before you get to mitosis. What's cytokinesis?
That's the last thing.
Yeah. Do you remember what we do in cytokinesis? So I'm going to show you something to where you'll never forget it. So even if you try. So cytokinesis. So kinesis means to split. Cyto means cytoplasm. So cytokinesis is the division of the cytoplasm. So this occurs after everything else because prophase, metaphase, anaphase, and telophase divide the nucleus, right? Divide the genetic information. But then cytokinesis occurs so that you have two separate cells. Okay? Awesome. Do you guys want to run through of what occurs in each phase, or do you guys feel comfortable about prophase, metaphase, anaphase, and telophase? You guys feel pretty good? Okay, cool. All right. Any questions about this lecture? All right, we'll move on. All right, great. So uncontrolled cell division is cancer, okay? Uncontrolled cell division can create tumors, right? Those are masses of cells. And so that is cancer. Inflammation is what we talked about in our immune system, right? Huh? Oh, okay. Oh, okay. Good. All right. Then I won't see more about it. Okay. Any other questions about any questions with this? All right. So all of those can cause a mutation. So what is a mutation?
Isn't it a mess with there?
Right. It's a change in our DNA, right? Do you think mutations will always be negative? Not necessarily, right? Awesome. Sometimes, often, mutations can be advantageous, and that changes the makeup of the population. But yeah, so it's a change in the DNA, right? And these are all ways that we can change our DNA, right? Smoking, sunburn, DNA replication, yeah. So just make sure that you know what a mutation is and what can cause it, okay? Awesome. Any questions about the cancer mutations lecture? Okay. So which process makes gametes? Meiosis, right? And so when we were drawing that chart, we draw out that meiosis makes gametes. So meiosis makes gametes. And what are gametes? They are sex cells. And sex cells are like your sperm and your egg, okay? Your sperm and egg, they must be haploid because in order for the sperm and egg to come in together and make a diploid cell, that means they both must have half the amount of information. So gametes are our sperm and our egg. And then they come together during fertilization, okay? Does that make sense? Okay, so just to clarify, so gametes are haploid, and then what would you do to make this sentence true? What would you say?
Somatic cells are haploid.
Yeah. Or gametes are haploid, right? Awesome, great job. All right. Okay. So, two answers are correct. Which two answers are correct?
Haploid.
And genetically unique, right? Because with meiosis, we're creating our sperm and egg, right? And all of those are different. That's why if you have siblings and you have the same parents, you're both different, okay? Because the sperm and egg that created you were genetically unique from each other and from your parents, okay? So meiosis makes gametes, which are haploid and genetically unique. Mitosis makes somatic cells that are diploid and genetically identical to each other, okay? There's a typo. The red one should say present in males and females. Okay. So one of you may have got red because I